Ahora. Okay, welcome to everyone, and thank you for your interest in our webinars organized by our RSG Colombia group, supported by the International Society for Computational Biology and supported also by RSG Turkey. Today we have the talk about genomics tools and applications for cannabis sativa by Daniela Vergara. Daniela, she's a microbiologist from Universidad de los Andes in Bogota, Colombia, and she holds a PhD in ecology, evolution, behavior from Indiana University, Bloomington. And nowadays, she's a research associate at University of Colorado, Boulder. So thank you very much, Daniela, for being here with us and for sharing this great talk. And please go ahead and thank you.
Well, thank you very much. Are you able to hear me well?
Yeah.
Hello? Okay. So thank you, Jacid, for having me. I am Daniela Vergara. I'm a researcher at the University of Colorado in Boulder. And I am the founder of the nonprofit organization Agricultural Genomics Foundation that aims on bringing cannabis science closer to a broad public. I'm also the co-founder of the Cannabis Genomic Research Initiative, which is our group at the University of Colorado. And my funding comes from Steep Hill, which is a testing company based in California, but we have many different facilities. And I've been working with Steep Hill for a little bit more than a year. And today I'm gonna be talking about research that has been done in cannabis and research that has been done. These are papers that I consider that have been really important for progressing cannabis research. And so I'm going to give you a big overview of my research, but also research from other groups. So this is my contact information, my personal email, my Colorado email. This is my Twitter, and this is my website. And so I want to start by thanking these people whose data I'm going to show. This is all collaborative research. Ezra Husher, Celine Feathers, Chris Pally, and our leader at CU, Professor Nolan Kane. I also want to thank our collaborators from CU Boulder, so Professor Kent Hutchinson from psychology and Professor Cinnamon Bidwell from the Institute of Cognitive Science. Our collaborators from the industry, from Steep Hill, Anthony Torres and Reggie Gaudino, especially, although we're a big scientific team and it's very collaborative. I also want to thank Ben Holmes from Centennial Seeds. Without him, we wouldn't be able to have data, and he's been growing several of our cannabis plants that we use for our research. And so cannabis is a plant that is a flowering plant, so it's part of the angiosperms. And it's a dioecious plant, so males and females are found in different plants, but there's hermaphrodites that exist. Cannabis is pollinized by the wind, so it's wind pollinated. The pollen travels through the wind. It's usually annual, so individuals live and die in one season during a year, but there's individuals that live in tropical and subtropical areas that may live more than a year. Cannabis is part of the family Cannabiaceae. This family has approximately 10 genera and approximately 100 species. And hops, so Humulus is the closest related extant species, so actually living right now is the closest related to cannabis. Cannabis has a genome size that it's about 818 million base pairs for females and 843 for males. It has 10 chromosomes, so 9 of them are autosomes, and then it has an X and a Y. So females have XX and males have XY, and the hermaphrodites have undifferentiated chromosomes, or what appear to be undifferentiated chromosomes. And the Y chromosome is much bigger than the rest of the chromosomes. One of the reasons that cannabis is so well known is because it produces a bunch of different compounds. These are some of the compounds that the plant produces. It produces terpenes that are usually responsible for the smell, and it produces cannabinoids that are the ones that interact with our human bodies. Two of the well-known cannabinoids are THCA, so tetrahydrocannabinolic acid, and CBDA, cannabidiolic acid. And these two compounds are part of this biochemical pathway. This is a complex biochemical pathway, and I'm going to focus on this part of the pathway. Although, so from here up, so all of this part occurs inside of the plant, and this part over here, this last part, occurs outside of the plant. So all of this part of here occurs in the trichomes, which are these little hairs found especially in the flowers of the females. And I'm going to focus on these three synthases. So THCA synthase, CBDA synthase, and CBCA synthase. So these three synthases use the same precursor molecule, which is CBGA. And if the plant has THCA synthase, then it produces THCA. If the plant has CBDA synthase, then it produces CBDA. And if the plant has CBCA synthase, it produces CBCA. This compound is produced in low quantities in the plant. And then when we heat the plant up, either smoking or vaping or baking, then we turn these acidic compounds into the neutral compounds, and those are the ones that interact with our bodies, right? So that's why we make brownies, or we smoke a joint, or we have a vape pen, because that's how they interact with our bodies. And THC is the molecule that is psychoactive. But we have to heat them up. So this part happens outside of the plant. And again, these three synthases, which are very similar, at least in their sequence, these three synthases, they use the same precursor molecule. Okay, so for cannabis, we have a genome assembly. A genome assembly is when we piece all of these little chunks of DNA together, kind of like a puzzle. So when we sequence a genome, we have to break all of the little chunks into small pieces, and then we have to put them together. Ideally, we would have an assembly with 9 autosomes and 2 sex chromosomes. So we will have 11 pieces, right? With all 843 million base pairs, million letters in the DNA. So that's what we're shooting for, right? We're shooting to have 11 pieces, and on those 11 pieces, they would add up to 143 million letters. And we would also have, ideally, a genetic map. A genetic map tells you where you are on the genome. It's kind of Google Maps, but for genes. So it tells you, I don't know, the gene for flowering time is found in this place over here. The gene for cannabinoids are in this place, and the gene for seed size are found here. So ideally, we would have also a genetic map, which we're in the process of making. We know more or less where are certain genes in the genome, but there's a lot to be researched. And so I'm going to go in chronological order on the papers that, in my opinion, are key papers for cannabis. So in 2011, we had the first genome sequenced. It was sequenced by a Canadian group led by Jonathan Page. Jonathan Page is the founder of Anandia Labs that got recently bought by Aurora. Aurora is probably the biggest cannabis company in the world right now. Aurora and Canopy Growth are probably the two biggest cannabis companies, both of them Canadian. So these guys in 2011 published the first genome. This first genome was a purple kush individual, which is marijuana type, so a drug type cannabis. The number of scaffolds was about 136,000. It had more or less 700 million base pairs. So it wasn't complete, or this genome wasn't complete. N50 was 16.2 kilobases, and the largest contig was 152 kilobases. And I'm gonna explain more or less these metrics, right? So N50 particularly, which is a measurement of, it more or less gives you how complete your assembly is. Contig is a continuous length genomic sequence, and scaffold is when you put these contigs together. These definitions I got from Google. And then scaffold N50, which I think it's an important metric, although it's a little bit, in my opinion, it's kind of iffy, a little bit, especially if you're reaching completion of your genome. But scaffold N50 is more or less the median. So 50% of the entire assembly is containing contigs equal or larger than this value. So half of the genome is in these contigs larger or equal than N50, or the sum of the lengths of all contigs of size N50 or longer, that they contain 50% of the genome sequence. So I got all of these definitions from Wikipedia, and this one from Molecular Ecology, which I think it's really, really good, and I'm gonna go through that one. So this I got, from Molecular Ecology. I really recommend reading this blog. They do a really good job at explaining complex things in this blog. So in this example, if the total length was 400 base pairs, half of the length is 200 base pairs, and that contig, so you organize all of the contigs into from largest to smallest, sorry. So this is the largest, this is the second largest, the third largest, etc. And then that one contig that gets you past half of the length is N50. So in this case, N50 is 60 base pairs. So for cannabis in 2011, we had a total of 787 million base pairs. So half of it's 394 million base pairs. And this contig over here of 16.2 kilobase pairs, that is N50. So this was in 2011, what we had in 2011. So then in 2014, we had this Russian group that published this paper. I have a lot of problems with this paper, but despite all of the little things, I think that overall it's an important paper to read. And what they describe is that there is this region on the Y chromosome that recombines with the X chromosome. And they describe the size of the chromosome. So the Y chromosome is the biggest chromosome of all, and then it's the X chromosome, and then all of the autosomes. So that's basically the take-home message from this paper. And they suggest that the region that still recombines is smaller, but we still don't really know how big is this region of recombination between the X and the Y, and we're still figuring that out. Then also in 2014, we had this paper published by Nora Volkov. Nora Volkov is the director of the National Institute on Drug Abuse, NIDA. NIDA is a subdivision of the National Institute of Health. So NIDA is a subdivision of the NIH. And they basically took the confiscated cannabis, so the cannabis from the black market, and they analyzed the potency through time. So what we can see in this graph for me is the most important graph of the paper. And despite the name of this paper, I think it's a good article, and I do not think that this title is reflective of the paper. So anyway, so what they show is basically that THC increases through time. So for me, what that means is that growers and breeders choose the cannabis with the most THC for those to be the parents for next generation offspring, right? And then probably the amount of THC, according to what I read from this graph, is a heritable trait. And therefore, if your parents have high THC, it's likely that you're also going to have high THC. And that's why we see this increase through time. And given that all of these enzymes that produce these cannabinoid compounds, such as CBDA or CBCA, given that they're so similar to each other, I think it's possible in the future to also increase the amount of both CBD and CBC. Then in 2015, we had a paper by this Italian Dutch group. So this guy, Etienne de Meyer, he works for GW Pharmaceuticals, which is a big pharmaceutical company based on the Netherlands. And they are the ones that produced Epidiolex, which has been used in kids with epilepsy. And it's a CBD-only product. So these guys published this paper in 2015, and basically the take-home message of this paper is that depending on the sequence that you have, you can produce either more THC or less THC. And this is a good table that I got from that paper. And let's remember that these three enzymes, these three synthases, use the same precursor molecule. And what we thought at the beginning was that at least these two synthases were two different alleles from the same gene. So we thought that there was only one gene that had these two different alleles, right? These two different forms of a gene, and they were codominant. So if mom produced CBD and THC, and dad produced CBD and THC, then the offspring, 1/4 would produce CBD, 1/4 would produce THC, and half of them would produce both compounds. So that's what we thought happened with papers from Mandolino 2003, De Meyer and Hammond 2005, and De Meyer 2009. But then in 2015, George Weinblain from Minnesota, he did a bunch of analysis with a back cross population of about 100 individuals. And what he found in this paper is that THC and CBD synthases, they are two different genes that are found in very close proximity. So these two different genes are found 1 centimorgan apart. So is everyone there? I want to know if people are there. Are people listening to me? Hello? I think I've been talking by myself. Is everyone?
No, it's okay.
Okay.
No,
there's people are talking and stuff.
Yeah.
Oh, great. So everything is okay. Okay. I just felt that I was kind of giving a monologue.
Yeah.
So and then let me know, Jesse, if people have questions, if I'm going too fast. You know, like this is what I do in my everyday life. So I know that things might be a little complicated, but But if you have questions, Jesse, please let me know if people are there. Okay, so I'm going to continue.
Can you hear me, Daniela?
Okay. So in 2015, George Weinblain from Minnesota then showed that these two genes are in very close proximity, but there's two different genes. So it's not one gene with two different alleles, but two different genes. And then in 2015, we also got this paper. Again, Jonathan Page from Anandya is part of this paper. And basically in this paper, what they found, which is not surprising at all for those of you who are marijuana users or who are involved at all in the marijuana industry, that the names of the strains. So first of all, people in the industry call them strains instead of varieties or cultivars. They call them strains, like bacteria or viruses. So right there, right off the bat, you know that the names are misleading. So basically what they showed here is that the cannabis strains are misnamed, right? And what people say in the industry is not entirely true, right? And someone may call, like there's this two big groupings, sativa and indica, which we still do not know whether they mean anything at all, but people call this is 100% sativa and this is 100% indica, and maybe they're super closely related. Or these two are 100% sativa and they're very distantly related. So those names really do not mean much. Okay, now I'm assuming that everyone in the public knows a little bit about biology, or we're all biologists. So we know that we have the chloroplast in the cell, right? So this is a plant cell, and here's the chloroplast responsible for photosynthesis, so basically making food out of light. And the chloroplast has a small genome of its own, and we have the mitochondria, which is the powerhouse of the cell, so it makes the energy of the cell, and this also has a genome of its own. So in 2015, we assembled the chloroplast. This is a hemp individual, and the chloroplast is a circular genome. So we got this published in 2015. In 2015 as well, Kevin McKernan, who is part of Medicinal Genomics, which is a group out in Boston, he doesn't really, he puts preprints out. I don't think that he publishes in peer-reviewed journals, but he put all of his. So Kevin McKernan put out this paper that basically says there's multiple genes related to the production of THC. So that was in 2015, along with the chloroplast genome. And in 2016, we got the mitochondria assembly. So we had the circular genome from the same variety that it was a Carmagnola hemp, so a hemp originally from Italy. And then in 2016 too, we got this other paper where we describe also the relationship between multiple strains. So this here, we have a split tree graph with approximately 200 strains. So in this split tree graph, what we see here is the relationship between all of these individuals. So the closest you are between each other, the more of the genome you share. So if you're very closely related, so these two guys, for example, they are very related to each other. They share a bunch of their genome, and here in this this thread-like structure that looks like a spider web is the part of the genome that's shared between everyone, so kind of the ancestral part of the genome. And what we see here, depending on how you look at this graph, is that more or less we have these three different groups. So we have the narrow leaf marijuana types, the hemps. So the hemps are the industrial hemps that are used for fiber, like that very strong fiber that's used for rope in boats, or that very strong fiber that's used for clothing. So that comes from hemp, or for oil. So it's industrial hemp, basically. And then we have the broadleaf marijuana type. So these two, the broadleaves and the narrow leaves, are used in medicinal and recreational settings. And this is the industrial hemps. And then we have individuals over here that are kind of the hybrids between these different groups. So that was a paper that was published in 2016. And basically my contribution for that paper, what I did was that I analyzed the chemical composition, so the chemotypes in these different groups. So we have here, I mean, this analysis has a lot of different caveats that I'm not gonna go in depth. If you guys have questions about it, I'm happy to answer. But basically what we can take from this analysis that I did was that the broadleaves have more THCA and that the hemps have more CBDA, basically. I also analyzed the other minor cannabinoids. For example, CBC, apparently hemp produce more CBC, and the terpenoids, right? The terpenoids are responsible for the smell. However, apparently, myrcene, for example, also interacts with our endocannabinoid system. So, and then again, these individuals, these different groups, for example, also differ in some of these compounds like linalool or terpinolene. So that was my contribution to this paper that was published in 2016. And then in 2017, we got this other paper where we review all of the available tools at that time for cannabis. And interestingly, this paper that was published, early 2017, it's already obsolete. There's already much more tools that we have available that we didn't have 2 years ago. So going back to these THC and CBD, when I was doing this analysis for THC and CBD, I wanted to look for the genes. Again, I wanna note that in 2015, McKernan and collaborators, they found out that THC synthase genes are found in multiple copies. And indeed, that's what I found. So here, this is the screen that you see when you have an alignment. So here is your reference. In this case, the reference was a THC gene, and then I aligned the genomes of multiple individuals. And what I found was that it looked like there were different chunks of the genome, different reads that aligned to the same genome, but they were very different from each other. So these reads over here, for example, they have, in this position, they have this T, these other over here have these other different sequences. So it looked like there were multiple sequences that were aligning to the same reference. So indeed, we found in 2 different assemblies, we found that there's different genes associated to cannabinoid production. So this is a paper that I currently have under review. And these are 2 different assemblies. One of them is constructed via PacBio. PacBio is a sequencing methodology where you sequence long reads. So you don't have to break the genome into small chunks, but you can sequence longer chunks. So one of them is sequenced via PacBio, the ones that start with 0. And then PK was that one assembly that was published in 2011 by Page's group, by Jonathan Page's group in Canada. So we see this is a genealogy from all of those genes found in those 2 assemblies. And we can see here that there are different clusters of these genes, that there's some genes that are related to the production of THCA, one from each assembly. So the green circles means that it's a complete gene. This hexagon red hexagon is kind of a stop sign, so it means that it's a truncated gene, so it does not make a protein. And what we can see here is that there's multiple genes, and that these two individuals, these two different assemblies, they differ in the number of cannabinoid genes. So indeed, we found that yes, there are multiple genes responsible for the production of cannabinoids. So there's copy number variation in these genes. And so we found that copy number varies within and between groups with the genes related to CBDA production. So here in the y-axis, in the vertical axis, we have the estimated number of genes, and we have our three different groups, and we can see that that individuals from the broadleaves or the narrow leaves, they have between 1 to 2 copies, and the individuals from hemp have between that have around 1 copy of the genes. And even though the median, which is this line over here, even though the median in the hemps is the lowest one, it has a bigger range. So this box, so in this graph is called a box and whiskers plots, and I really like them because they show how the data is distributed. And what we can see is that the hemps have a lot of a big range of distribution in the number of copies related to CBDA genes. And same thing for the THCA genes. We can see that the broadleaves and the narrow leaves have around 1 to 2 copies, or even some of them may have even more than 2 copies of the THCA related genes, and the hemps have approximately 1 copy of those THCA related genes. So indeed, we found copy number variation within and between groups for these cannabinoid genes. So now in 2017, so going back to our timeline, in 2017, we got this other paper published about the cannabis produced by the federal government, produced by NIDA, the National Institute on Drug Abuse. NIDA, again, Nora Volkov, whose paper I showed in some previous slides, Nora Volkov is the director of NIDA. So NIDA, which is part of the NIH, the National Institute of Health, NIDA produces the cannabis that scientists must use for research, right? So scientists might use NIDA's cannabis for research. And again, NIDA, the National Institute on Drug Abuse, is part of the NIH. So I just want to let you know how the government-produced marijuana, the one produced by NIDA, this is how it looks, right? And this is the cannabis that we can get in dispensaries here in Colorado. If you're over 21, you can buy this cannabis. And if you're a scientist, you must use this cannabis. So, Steep Hill, which is the company that finances my research, Steep Hill, they test for the chemotypes of the different cannabis. And by law, if you have a dispensary and you're selling cannabis, you must test your cannabis. So this is what Steep Hill gives their customers. They give, your cannabis produce this amount of CBN, CBG, etc., and this amount of terpenoids. So this is an example of what Steep Hill gives to their customers. So what we did is that we went into NIDA's website and we downloaded all of the chemotype information, all of their chemotype information for their cannabis, and we compared it to that cannabis that was tested by Steep Hill. And we had, again, so I wanted to show you. So again, we compared this cannabis to this cannabis. And so we had 4 different locations for the private markets with data from Steep Hill. We had Seattle, Oakland, and Sacramento, Denver, and the one produced by NIDA, which is produced at the University of Mississippi. So the University of Mississippi has a contract with NIDA, and they are the ones that grow the cannabis for scientists to study. And so it's one guy, Mohammed El Solhi, who grows all of the cannabis used for medical studies and for scientific studies. And he is here in the University of Mississippi growing this terrible weed. So, okay. So what we can see here in the vertical, the y-axis is the percent cannabinoids. We have this first cluster, we have CBD, the second bar cluster, we have THC. And what we can see is that NIDA, which is this second bar in red, orange, is the one that produces the least amount of CBD and the least amount of THC. And I erased the one-to-one comparisons. You can find the one-to-one comparisons in the paper, but basically NIDA differs from everyone else in the production of THC and differs from everyone except for Seattle. So Seattle and NIDA are not different in the production of CBD, but all of these other places are different in the production of CBD. Now here we have a principal component analysis, and the principal component analysis basically tells us the overall variation in cannabinoids. So if we have this overall variation in cannabinoids, we can partition this variation using a k-means clustering analysis. We can partition that into two, this gray group over here and this pink group over here. And what we can see is that NIDA is only part of one of the groups, right? It's part only of the pink group. And if we partition all of this variation by location, what we can see is that Oakland in blue and Sacramento in purple, they contain NIDA's variation, but NIDA does not contain the variation found in Oakland and Sacramento. So basically, NIDA underrepresents the overall variation that we see in cannabis chemotypes, right? In all of these different compounds that the plant produces. NIDA only contains a fraction of that overall variation. So in 2018, we had this paper, which is a review paper in this very prestigious journal. And we had this paper that was published, and it said that cannabis, so they reviewed all of the literature, and they say basically that cannabis is useful for the treatment of three things: pain in adults, nausea and vomiting induced by chemotherapy for those patients with cancer, and spasticity in multiple sclerosis. So only those three things. But again, the studies have been done with that cannabis produced by the government and not with the cannabis that people are actually consuming. So most of the studies have been done with NIDA's cannabis that underrepresents the overall variation that we see in cannabis. Okay, so now continuing with our timeline. Now in 2018, Weinblain, again, the guy that discovered that the THCA gene and the CBDA gene are two different genes that are found in close proximity. So Weinblain joined forces with these guys from Sunrise Genetics, which are actually collaborators of ours too, and they did an assembly with the sequences that Weinblain had before with that backcross that Weinblain had. They sequenced it. They did a much better sequencing job, and they found that all of these genes, all of these cannabinoid family genes, THC, CBD, and all of these other genes that we don't know what they're doing, they found that all of these genes are in very close proximity and that they're full of transposable elements, so of junk DNA all around them. So that they're very close in proximity, and they have a really good assembly, which is publicly available. And in this assembly, they have 10 chromosomes. They do have about 200 other scaffolds that they did not don't know where they belong in the genome, but they have a really good assembly. At the same time, also in 2018, Kevin McKernan also released another assembly. His paper, all of these two papers are preprints. They are not published in peer-reviewed journals. They are publicly available, and their data is also publicly available. So McKernan released this genome that also has, this one has about 300 different scaffolds. So this other one is a little bit more complete. In that sense, in that this one has, like 10 chromosomes and 200 other chunks. This one has 300 different scaffolds. And what he also found is that all of these genes are in very close proximity. And again, in 2018, Jonathan Page's group with Van Bakel, who were the ones that released the first genome, the Purple Kush genome, they redid that Purple Kush genome, and they have, this paper was actually published in a good journal, but in my opinion, these two other assemblies are much better, despite this being in a peer-reviewed journal. And the problem with McKernan's assembly, I've played with these two assemblies. I've been working with these two assemblies, and the problem, in my opinion, with McKernan's assembly is that he has gotten several iterations of the assembly, and he has done it several times. So it's really hard to keep up with which assembly is the last one and which is the one in NCBI. So if any of you are going to use this assembly, I recommend just sticking to one, like go to NCBI, download the one that's in NCBI, which might not be the latest assembly, but at least you know that that's your go-to, because then he has another iteration that is lacking some genes, and it's kind of very disorganized. So anyway, so that happened in 2018, and then in early 2019, so early this year, there's this company that's, I think it's based in, I think it's Israeli, but they're based in the Netherlands. I'm not entirely sure, but there's some people from this company that are here in the US. At least some of them are in Michigan, I think. And Energene, they claim that they have a really good assembly, better than the others' assembly. So Purple Kush, this is Jonathan Page's, Jamaican Lion, this is Kevin McKernan's, and this one is from Grassan, the Sunrise Genetics people. So these two assemblies are the ones that I've been working with. And Energene, they say that they have a diploid assembly, so not a haploid assembly. So basically they have two different chunks for, like one coming from mom and one coming from dad. It's kind of confusing to me, because if you don't have your complete assembly, how do you know which chunk comes from each parent? It's kind of confusing to me, but they say this assembly, to my knowledge, it's not publicly available. So this is what they claim, and this is in their website. They also had this very pretty brochure that they distributed in the Plant Animal Genome Conference in early January. But in any case, they say that these are the metrics for all of the assemblies, and they're comparing all of the assemblies to theirs, and these two are the ones that I've been using. So these two assemblies, I don't know why they did not. This is not that hard to calculate what is the maximum scaffold length for Grassas and Busco. So Busco are the single copy ortholog genes, and the higher your Busco number, then the more, it's another measure of completeness. So they claim that theirs is better because they have more Busco genes than the other ones. But in any case, if it's not publicly available, then it's useless, in my opinion, or at least for me, because I cannot work with it. So anyway, so I've been working with these two assemblies, and what I did recently, for the reviewers from my paper, asked me to use these two assemblies to look for the cannabinoid genes, and I did. So for Grassas assembly, I found 12 genes related to cannabinoid production. These genes are in the same chromosome. And then for occurrence assembly, I found 27 different genes, which are in 7 different chromosomes. And I found one gene that is very likely the for CBD production in one of the assemblies, and another one, and which is likely the CBC production. I did this one with 97% for THC. I am not entirely sure it's the one responsible for producing THC. And then for occurrence assembly, I found one gene that is highly likely CBD. Another one that's highly likely CBD. So these are results that I've been getting lately. This is the research that I've been doing lately. And then I took a bunch of different individuals, including 2 NIDA strains, so 2 strains from the National Institute on Drug Abuse. We got DNA from those strains somehow, and we sequenced them, and so we aligned them to Grassa's assembly, and this is how the alignment looks like. So it's supposed to have a Poisson distribution. And why does it have a Poisson distribution? I recommend these websites that explain why, when you have an alignment or an assembly, why should it have a Poisson distribution. I found, I think it was this one and this one, these last 2. Oh, actually, Illumina's explanation is actually very good. So I recommend these websites. And with that alignment, we found, so in general with cannabis, the problem with the assemblies is that cannabis has a high heterozygosity. There's multiple alleles that are mistaken for genes. So you have alleles that are very different from each other, that are very heterozygous, and therefore you think that there's 2 different genes when indeed it's one gene that has multiple alleles. So it's been misassembled. So you place those alleles in different locations thinking that they're different genes. And the genome of closely related species, for example, hops, so humulus, they're in a even worse state. Some people claim that the beer companies, so that the breweries, the big breweries, I don't know, Coors, which is actually here in Colorado, that they have good genome assemblies, but they're not publicly available. So again, it's like they're non-existent, at least for me. Anyway, with those genomes, what I did is that I constructed this split tree graph, and these are preliminary results. And this is the paper that I'm going to be working on in the near future, which is kind of the continuation of the chemotype analysis from NIDA strains. So now I'm doing the genotypic analyses. And what we find here is that NIDA strains, again, are super close together. So basically, these 2 strains underrepresent all of this variation that we see, right? So NIDA is making scientists study this very little amount of variation when we have all of this variation that we can study. And we see that these 2 NIDA strains are actually close to the hemps. They're not what people are consuming, which are mostly here or here. So that's what I'm working on as we speak. And with that, I want to give 3 pieces of advice to people in the cannabis industry or consumers. This was from a presentation that I gave recently. So first, do not guide yourself by the names because strains are misnamed. Second, be more flexible with the amount of THC because this is a hypothesis that I have because enzymes may be sloppy. And 3, grow the most amount of diversity, including industrial hemp. So this first advice is for consumers, particularly the medical consumers. The second advice is mostly for legislators. And this third advice is for growers and breeders. So strains are misnamed. We go back to our split street graph published in 2016. I'm gonna zoom into the narrow leaves, and we can see that we have these two Durban poisons, or these two chocolates, or these two crypt out cookies that are probably the same thing. They're closely related, they're close on the graph, but we have these two Jack Herrers that are more closely related to something else, this one to Golden Goat, this one to XJ13, and then we have here these Girl Scout cookies, and if we zoom in to the other side of the graph, we have these other two Girl Scout cookies and these other two kosher kushes that are related to something else entirely. So, and then here in Colorado, hemp, the legal definition of hemp is whatever produces less than 0.3% THC. In places like Colombia, where I'm from originally, in Colombia, it's less than 1% THC, but that's a legal definition of hemp. But actually, this variety over here, this strain Otto, which was produced by Ben Holmes, who's our collaborator. So Otto has less than 0.3% THC, but it really does not cluster with the hemps, right? So maybe these legal definitions are also helping for the misnaming, which was something that was discussed by Soler et al. 2015. I talked about this paper earlier, and about and us in 2017, and I talked about these two papers earlier. So when in the industry they are talking about indica and sativa and the popular strains, that's very questionable. These popular strains are probably misnamed, and these groupings are probably misnamed. So consumers are probably not getting what it's advertised, and this is problematic, especially for medical patients, and growers and breeders and dispensaries might not really understand what they're producing or what they're selling. And so my recommendation is that testing strains should be mandatory. So in other words, if you go to a dispensary and you say, hey, I use this particular strain that has this particular chemotype, and maybe in that dispensary they'll tell you like, hey, I have this strain that has a very similar chemotype, it's called Danielita's weed. So you base yourself on the chemotypes and not on the names. Or you use closely related strains. So for example, you use Wonder Woman for your back pain. So instead of using Wonder Woman, you find, I don't know, Skunk Number 1 or Jack Flash that are very closely related. So the second advice, more flexibility with the amount of THC, especially for legislators. I think that these enzymes could be sloppy. In Colorado, you have to have a limit of less than 0.3% THC, which is very low. Again, these three synthases, they work on the same precursor molecule. So my question is, and this is a hypothesis, I don't know if this is true, but my question is, could THCA synthase produce CBDA? Or could CBDA synthase produce a little bit of THCA, right? Are these enzymes indeed sloppy? And if they are, then we should have more flexibility with breeders and growers, right? And increase, maybe my recommendation is to increase the limit of THC. Really, no one is gonna get high with 5% THC when we have strains that produce more than 25%, or we have concentrates with 98% THC. These levels, so these numbers, the 0.3% or the 1%, have not been proposed based on scientific data. These were legislators that I don't know where they found these numbers. And really, we do not know how much THC is it necessary to be psychoactive. Does it vary from person to person? What makes me high, is it also gonna make you high? We don't know that. There's no research so far on those things. And then advice number 3, grow the most amount of diversity, including industrial hemp. And this talk I gave in Colombia, so including varieties that have been grown there traditionally. In Colombia, we have, like mangoiche, Punto Rojo, Santa Marta Gold, which are the, classic strains that are probably adapted to the environment, for example, the humidity in Colombia. And we calculated pi, which is sequence heterogeneity. And pi, basically, we count the number of letters that differ from sequence to sequence. So in this one, this second sequence differs from this one in this C. This other one differs in this TCG, and we count the number of base pairs of differences. And for these three groups, we estimated pi here in the y-axis, and we can see that pi varies from 2 to about 3% for the three different groups. I am not entirely happy with this graph. These are preliminary results, but basically what this suggests is that cannabis have 2 or 3 times as much diversity as all of these species that have less than 1%. So as bonobos, chimpanzees, humans, thyme, or domesticated corn. So it has much more diversity. So in 2 full siblings of cannabis, 2 full siblings, you can find more diversity than you can find between me and that chimpanzee, right? Between me and that chimpanzee, there's less diversity than maybe 2 full cannabis siblings. So there's a lot of diversity. There's other species that have the same amount of diversity, but basically cannabis has a lot of phenotypic variation. It can be allowed, so this allows it to be used in multiple settings: medicine, fiber for rope, clothing, biofuel, beauty products. And therefore, if you grow the most amount of diversity, you might have genes that are very important for the conditions that you're growing in. For example, maybe disease-resistant genes or some other cannabinoids that we don't know about. So that's my advice: grow the most amount of diversity. And with that, stay tuned for more research. Again, this is my contact information. And if you can, please donate to our nonprofit organization, Agricultural Genomics Foundation. And with that, I'll take questions if there's time. Thank you.
Hi, Daniela. Can you hear me? Hi, Daniela. Hi, Daniela. Hello.
Okay, so can you hear me? So 2 questions. So Osman asked 2 questions. One, couldn't hear your explanation about the hermaphrodite plant. So my question is, what are the genetic bases that explain the hermaphroditism in cannabis? What is known about hermaphrodites in terms of THC and CBD production? That's a great question. We don't know how hermaphrodites are produced. I'm gonna go back to one of the slides. So this one from Divashuk et al. 2014. We know that there's recombination going on. We know that probably hermaphrodites. So when you stress a plant out, so when you have a female plant and you stress it out, for example, with silver nitrate, and this is something that breeders and growers have been doing for forever, or even with the sun. There's some individuals that produce male flowers, or some male individuals that produce female flowers. So we think that perhaps, the regions that determine sex are in the pseudo-autosomal region, or maybe even the regions that determine sex are on the autosomes. We don't know that. We think this process of becoming dioecious, right? So from hermaphrodites to dioecious, this process is ongoing as as we speak right now in cannabis. We think that this is a very new ongoing process. So we really do not know about the sex determination in cannabis. However, most of the individuals that are hermaphrodites are hemps, right? So in hemp, we find a lot of individuals that are truly hermaphrodites. Flowers in one plant. And those individuals are used in industrial settings. So those individuals are not used for THC and CBD. So usually the hermaphrodites, and in the industry, in the cannabis industry for recreational settings, when you see a male and when you see a hermaphrodite, they take him out. So usually the hermaphrodites are in hemp. Do you guys have more questions?
Can you hear me, Daniela?
Q&A. Oh, Q&A. No questions. Chat.
Okay, Daniela.
Yeah, Osman, is that a little better? Does that explain a little better?
Hello, hello.